You're famed for your choice of C. elegans as a model organism, and I know you've written about that too. What were you what were you looking for in an organism, an experimental organism, that led you to C. elegans?
The first thing we were looking for is to solve a finite problem. What we knew from the old literature is that the number of cells in these nematodes was quite small. That meant that if we were going to do things, we want to know everything, you see. Now, as you know, Isaiah Berlin wrote a thing called The Hedgehog and the Fox. And roughly speaking, you know, the hedgehog knows a lot about one thing, and the fox knows a little about everything in the old fable. But I decided the organism I wanted to be is the octopus, which has to know everything about everything. But in fact, it becomes very important if you're tackling a next level of complexity. Once we got away from single-celled organisms, we had to face the challenge of taking something which starts as one cell, develops a whole lot of different cells, and which is the essence of what we need to explain at this level. And therefore, we need something that we can accomplish a finite thing. So, for example, by sitting down and spending 20 years and working out the wiring diagram, how all the neurons are connected in this animal, what we wanted to do is to be able to say, if we had a theory to say, well, you see these neurons, this is the way this works. I always imagine the skeptic who says, how do you know there isn't another neuron, you know, that goes down the back of the body, through the big toe, comes up the front again, and that's doing everything. So to answer him, you have to say, there are no more wires. We know all the wires. How do genes control the construction of cellular machines that generate behavior? In other words, we want to know, how do you build a neuronal network? And the second thing is, how do these generate the actual behavior, which is at the next level? And what are the problems about that? And we knew that there was a midpoint here that we had to get strict descriptive information. And that was the structure of the nervous system.
I remember a quote of Chargaff's where he's discussing DNA sequencing, and the phrase is something like, not in our lifetime will we be able to sequence DNA.
Of course, you see, we believed right from the very start, from the day, the very moment I saw the model, I said, we have to know the sequence. And of course, today, if you were to ask a student, how would you show that these are related? He would say, well, of course, I'd just get some DNA and I'd send it out to be sequenced, and I'd get the protein and I'd give it to someone else to be sequenced, and then I'd see how they were matched to each other. So it's trivial to do that today. And, but I think in the early days, it posed a gigantic challenge, which I think was met by the really much more clearer idea of how to do genetics, basically.
Let me just stop you a minute. You mentioned going to see the model. So we ought to just say very briefly what you remember of that moment.
We went over, and I landed up meeting Francis and Jim for the first time and seeing the model. And to me, that was it. You know, once you saw that, you knew that's, you know, this genetic code must be solved, the whole mechanism must be solved. There's nothing else in biology. And what was interesting was that, of course, most biologists didn't believe it. In fact, I believe that for about 3 years after the model, nothing happened. And it took the 56 things began to happen. But of course, within the next 5 years, it was all over. So there was this incredible period, reaching culmination in the early 60s, late 59, where everything just became a new way of talking about things.
So, coming back, in a way, what we were talking about at the beginning, the style of research. In this period, I mean, I think you were on a paper with 3 others, the general nature of the genetic code.
That's right.
That seemed to be about the sort of maximum number of people that appeared on any paper about that time.
Well, I think it was, we wrote all that up because that was with Francis, myself, and we had 2 other people listed on that because they contributed to doing this. But I think that that was the maximum. But we had a rule, which, I mean, Francis expressed very vehemently at times. That is, nobody goes onto a paper unless they've either made a contribution to the actual work or contributed an essential idea. And the definition of an essential idea was that it was not the magnesium concentration. So that's why it's a joke.
Yes.
And I think that it was the essential thing. And we didn't have this putting on the chap who runs the lab onto every paper. That was not done. And so all the people who contributed to the thing were on the list, and the list was, and Francis was very strict about this. We had no freeloaders. Well, as you know today, there are only 2 important positions on a list of authors, some of which could be 100 long. There's the first author, who's probably done all the work. There's the last author, who's the head of the place and raised all the money. And there's all this region of anonymity. And I call it the caboose theory of publishing.
Sydney, I thought we might pick up on what we were talking about last night, where we were discussing the nature of modern experimental biology research, in particularly the large-scale, non-hypothesis-driven types of research. And you have some rather strong feelings on that sort of thing, projects like the ENCODE project and so on.
So whereas originally, and I can remember that grants were given for what we used to call a boy and a toy. That is, you gave a grant, you had one assistant, who was the boy, and you had a toy. They gave you you got a pH meter. That was a sort of standard start-off grant. Now these had become very large as well, and groups began to grow in sizes. So, 30 was not uncommon, 60 was becoming common, and this now meant that most active scientists had left the field and had become managers. In other words, they took early retirement at about the age of 32 at that stage, because they had now had problems, which was to fuel the grants. The grants needed fueling with results. And then, of course, when the money came, they had to hire more people to get more results. So these things grew. And of course, we're now in the middle of quite a crisis in the funding of basic biology. And I think it's good to look back at the past and see, exactly how did it change. And of course, if we look at what has happened in the last 60 years, we've just been living in a period of constant revolution. That is, every decade, something happens which now enables more things to be done and better things to be done. And I think many of the things that we thought we'd never see in our lifetimes in terms of information are now realizable quite easily.
Do you think this big science approach, the multi-center, hundreds of people approach, in a way is done because in a way it's simpler? You simply say, we're going to do an analysis of we're going to do some big approach like ENCODE, or we do some big approach like the Human Genome Project. In a way, you don't really have to think about particular experiments. You just go in and produce all the data.
I think that those things are when it's clear, you see. I always liken, as they did at the time, the Human Genome Project for sending a man to the moon. And I always thought that was an apt relation. In fact, it was a huge technical accomplishment, sending a man to the moon. And of course, they were all estimated to cost about the same, about $3 billion, as I recall at the time. You see, but it's apt in the following way, because sending a man to the moon is easy. It's bringing him back that creates all the difficulties. And that's the thing. So I view the human genome as stranded on a moon somewhere. We knew right from the start that the interpretation of the sequence would be the important thing. And in fact, several years ago, I realized that nobody knew anything about the human genome sequence except a bunch of computers. So there's no reason to believe that if you sequence people with cardiac disease, you're going to find a whole lot of mutations. And I think one of the interesting things is the following. People have been doing this and looking at brain, neuron, brain retardation, autism, all these rather complicated behavioral things, which are almost certainly developmental in origin about the brain, you see. And so they finally will locate the piece of DNA which has either been deleted or duplicated, and they then write in their papers, oh, we found on this piece the following genes involved in the brain. But half the genes are involved in the brain, So likely wherever you land. So there's no way we can at the moment implicate any causality. And I think that this is now just a total waste of time. You'll find people, sometimes we find clusters. There are things we have to explain, but they're not in the current way of talking about genomes. So I believe that that we should still allow people to look at these genomes and analyze them very carefully to see if we can do this reconstruction as a part. Of course, it's completely valueless information until it'll prove to be valuable.
Do you think that the machinery and the neural brain structure is sufficient or will provide an explanation for what we experience as consciousness?
Yes. Well, I think that the first thing I want to say with this, of course, is there is a belief that, of course, the mind is not separate from the brain. I mean, that's the first thing. And the one thing that becomes very interesting is, as you know, in discussions of consciousness, there has been a discussion of Turing's paper in Mind. Now, what I hadn't realized till quite recently is that he called it an imitation game. It was not a simulation, you see. In other words, he was saying to you, I'm going to lock up in a room. In one room, I'm going to put a computer, and in one room, I'm going to put a human being. And you're only allowed to ask them a certain range of questions. And can you distinguish? Can a computer imitate a human being enough to con you, to actually do this? So it was called the imitation game. That's not the game we're interested in. I think that's doable. There was a very famous program called ELIZA, which imitated a kind of psychiatrist confidant, which a lot of people loved, but it was completely silly because it just turned your question around, your statement around. For example, one piece of the conversation was, my boyfriend bugs me. The computer replies, does it bother you that your boyfriend bugs you? And when it got lost for how to follow up the conversation, it then said, you haven't mentioned your father or mother yet.
Sounds like a very good psychiatrist.
It was a very good psychiatrist, but it had nothing. And clearly it's not a human being there, but a very clever way of doing it. That's why I always think I could easily write a computer program to talk to royalty, because you're not allowed to initiate new topics when you talk to royalty. Anyway, besides that. So now let's look at this problem of consciousness. What I believe is that consciousness will not be solved in the way it's being put now, but it'll disappear. So I think the idea that there'll be some special part of the brain devoted to this, and that we'll finally find the seat of consciousness, that was, I think, is going to be misplaced. And I believe that in 50, 20 even years' time, people will look back and say, what were they talking about? Which I think is probably the most exciting thing about brains, is that even things we think are really fixed, by our genomes, have to be learned. In other words, the brain is essentially unfinished in most animals, higher animals. You get above a certain level of complexity. It then, and this depends on very interesting observation made by Hubel and Wiesel, very famous one, in which they showed that the brain wires back correctly, but if at a certain period in development, very early development, they closed one eye, the animal became blind in that field. It's called ocular dominance. And it was only seeing the eye which was really being... So we need to match the function of the neuron to what is on the outside. Again, then it's successful. So I think what we have is an unfinished thing, and then we learn to use it in a way that it matches our environment. And I think there's a most remarkable, simple case which we can all study and which hasn't yet been done properly. And that is, you can train muscle in a test tube. You can train it, okay? And I think that's where we start. And we know what it's all got, we know what it's got to do with. It's got to allow calcium to enter the cell. And I think all of synaptic learning, of muscle training, is all that fundamental mechanism, which is, okay, we left an unfinished brain, finish it off the way it suits you.
So what do you think of this proposal for the brain activity project, or I'm not sure what the correct title is, that's being proposed?
Well, I think it's a good thing. But I mean, for what they want to do, which is, let us say, record from 1 million neurons, we're not going to be able to do that very easily with human beings. So I think we'll have to learn by other processes. That's why I think that the brain part, which is hardwired, okay, which is called the hypothalamus, which has nothing to do with consciousness, okay, that's something that we can do because it's much more invariant, same as many sensory organs. So I think if we can work out how those work, and we can learn how that all evolved. See, and I think there is remarkable information buried in the human genome. It's actually buried in all the genomes of extant mammals, of how these things evolved, and in the past. And I think that that's become, at least my objective is to extract that, and in order to see, to solve the question of how did the eye develop. Because I think the eye developed from something that smelt. In fact, I can prove it. I think we'll see a huge breakthrough in the whole of this area, which we could call human biology. I think there will be incredible things learned in the next. I'm just sorry not to be able, that my tenure will expire before that. But I think this would be the complete apotheosis, of starting with this tin plate model 60 years ago.
Thank you very much, Sidney.